lecture. So hence, I request Dr. G.R. Janardhana, and also I thank Dr. Rajkumar Garampalli for giving him the opportunity for to interact with the upcoming professors joined here for refresher course on the important theme, the bioresources for sustainable development in the modern era. Sir, over to you, sir. Thank you very much, sir. Thank you very much.
Thank you very much, Dr. Amritesh, for introducing me. At the outset, I also thank the HRDC and the coordinator of the 16th refresher course in life sciences, Dr. H. Rajkumar Garampalli, for having kept my name for the today's second session to address the participants of the 16th refresher course in life sciences. You must have been already familiar with various aspects of life sciences and the recent advances that have been made in the field of biological sciences. Hope after attending this refresher course, you will get back to the regular mode of teaching and where the learned knowledge will be shared with the fellow students to enrich their student knowledge. So the topic that I have selected for today's presentation is on infectious RNAs. It is one of the important current research topic in biological sciences. So therefore, we have also done some basic research work in our university. So therefore, I will be sharing some of my experiences with infectious RNAs, that what we did here, and also what exactly, how these infectious RNAs are going to rule in the 21st century. Let me share my presentation first, and then are you able to see the presentation?
Yes, sir.
Yes, sir.
Yeah.
Yes, sir.
Yeah.
But it should be more to slideshow, sir.
Okay. As you know, the world is bracing for the climate change. India is not an exception. And though Indian agriculture is an important backbone of Indian economy, earlier 70% of the population were dependent on Indian agriculture. And today, the 42% of the rural population depends on agriculture. Though the percentage of population involved in Indian agriculture reduced, India is the world's largest producer of many fresh fruits and cereal grains and vegetable crops. And Indian agriculture contributes about 20% of the gross domestic product. And total food grain production in India is estimated at 314 million tons of food grains. It was during 2022 to 23. It is all because of the green revolution that we have successfully launched in our country to end the hunger and malnutrition. So, though we have successful in achieving the green revolution, the future of green revolution and the sustainability depends on the evergreen revolution. We cannot keep quiet here. We need to do a lot of things to double the farm production. Then only we can achieve the evergreen revolution, because the climate change is affecting our crop patterns and affecting the world food bioresources. Therefore, it is very important that we need to look for evergreen revolution to feed this growing population. India's population will be 1.6 billion by 2050. How to feed this growing population? The world population will be 9 billion by 2050. So it becomes a very big challenge for agriculture scientists, because there are many biotic and abiotic factors reducing the crop production all over the world, so also in India. We are dependent on 20 to 25 important major crops for our staple food production, and we need to harness them. Among the important crops that we have that we have domesticated, potato is one of the staple vegetable food crop. Potato is one of the staple vegetable food crop in the world. It is native to America, domesticated first in Peru, and there are about 5,000 varieties of potato all over the world. And actually, Spanish introduced this potato crop to Europe in the second half of the 16th century. And because of favorable weather conditions in Europe and also in Brazil, Peru, made this crop as an important vegetable crop also in Europe and rest of the world. So also it was introduced to Asia and other countries. And today, China is the number one potato producing country, followed by India. It is stood in the second place. If you remember the Irish famine, and it was caused by the late blight of potato disease. It was in 1842 to 45. The late blight disease in Ireland and also in other parts of the Europe was completely devastated by this late blight disease. The fields promised bumper harvest. They were all transformed into rotten fields within a span of 15 days, and tubers were also spoiled underground. The Irish people, they dependent on only this crop as a staple food crop. They were out to suffer from hunger, dysentery, typhus, and 1.5 of the Irish people died from hunger and related diseases. During the same period, the British royals bought an eviction act, and the tenants were thrown out of their house in Ireland for not paying the rent. Finally, the help came from the United States of America and also Canada, and millions of people left to homeland Ireland. The Ireland population, which was once 14 million, reduced to 7 million due to this late blight disease. This is the picture just to show that tenants were thrown out of their homes in Ireland for not paying the rent because the British royals brought an eviction act. And homeless families, they took shelter on countryside. As I mentioned, 1.5 million people died, and almost same number of people migrated to other parts, particularly USA and Canada. It was such a serious famine, and which was responsible for Irish famine of 1845 to 49. It was in as a memorial was created in Dublin for remembering this great famine, which saw the population of the country halved through death and emigration. It took almost 15 years. It was in 1861, Anton de Bary, a German botanist, he did a simple experiment that proved that late blight of potato was indeed caused by a fungus, and he named it as Phytophthora. That means a plant destroyer responsible for the disease. That was the beginning of plant pathology. So Anton de Bary, this German botanist, he is called as father of plant pathology. Today, the discipline of plant pathology has diversified, and it has identified the various aspects of plant diseases and causative agents of plant diseases, mechanism by which plant diseases are caused by different biotic and abiotic agents, and methods to manage them effectively to protect the food from ravages of diseases and pests. There are various kinds of pathogens which cause diseases in plants. All these plants, the pathogens, can be classified into non-parasitic diseases, which are otherwise called abiotic agents, and we have parasitic diseases, which are called biotic agents. So abiotic agents or non-parasitic diseases include mineral excess deficiencies, toxic chemicals, toxic pollutant, extreme environmental conditions that as temperature, relative humidity, and moisture, air pollutant, excessive pesticide, excessive fertilizers, all these comes under the non-parasitic diseases which are responsible for causing diseases or deficiency of crop plant diseases. Apart from these, we have many parasitic diseases where the biotic agent such as bacteria, fungi, algae, protozoa, angiospermic parasite, nematodes, insects, phytoplasmas, viruses, and now virides are also known to cause diseases which take away 45 to 50% of the agriculture produce in a countries like India. Among these, the viruses are the very important submicroscopic nucleocapsid particles. They are very important causative agents of plant diseases. As you know that viruses do cause plant diseases just like animal diseases. They are made up of nucleocapsid particles, consists of a small piece of nucleic acid, either DNA or RNA, surrounded by the protein coat or the lipoprotein capsid. Their nucleic acid size ranges from 1000 to 250,000 base pairs. All these viruses which cause plant diseases, they are ultramicroscopic. We can visualize them only under electron microscopes, and they are acellular in nature. They does not have cytoplasm. So therefore, we consider them as acellular in nature. They are non-culturable and inanimate media, so very difficult to culture them. And that's why they are also considered as obligate intracellular parasites. It was Adolf Meyer, a Dutch botanist in 1886, for the first time identified the association of plant virus with a plant system. Later, it was a Russian botanist D. Ivanovsky stated that the viral diseases are infectious in nature. Later, it was M.W. Bejerny, who in 1898, he called that infectious nature of the virus as living infectious fluid, and he coined the term virus. Virus means a poisonous fluid. It was W.M. Stanley in 1936 first purified the virus, tobacco mosaic virus, associated with tobacco mosaic disease, and it was a major breakthrough as far as virology is considered. The systematic study of virology began. For convenience of our understanding, so there are David Baltimore, a well-known virologist, classified viruses on different contexts. I would just to use the simple classification system of virus. All viruses can be classified into animal viruses, in which DNA is the genetic material, and we have plant viruses where RNA is the genetic material, and we have bacteria infecting viruses are also present where DNA is the genetic material. But there are exceptions are there, like HIV, wherein RNA, though it is an animal virus, RNA is the genetic material. Similarly, plant viruses like Gemini virus, though in plant viruses, RNA is the genetic material, but in Gemini viruses, DNA is the genetic material. Similarly, we have MS2 and Q beta have RNA as genetic material. So this is an animal virus. Example for animal virus, a nucleic acid externally surrounded by a protein coat. In turn, this protein coat is surrounded by a lipoprotein membrane. There is an HIV virus. So we have a RNA, a nucleic acid, externally surrounded by a protein coat, and finally it is surrounded by a lipid membrane. We have a plant virus like tobacco mosaic virus, centrally placed RNA, externally covered by a nucleocapsid protein. Capsid, it is otherwise called capsid. It is a bacteriophage, centrally placed DNA, nucleic acid is the master molecule, externally surrounded by a protein coat. This is the characteristic feature of all viruses. But no one has ever imagined that nucleic acid alone able to cause diseases in plants. Scientists in the 20th century, they proved that just a small piece of RNA, smaller than viral RNA, able to cause diseases in plants, and they called these as infectious RNAs. Dear friends, today the topic that I have chosen is about infectious RNA, the molecular parasites of crop plant, on which I am going to speak for about another 45 minutes to 1 hour. And these infectious RNAs have been otherwise referred to as virides. These are the names. Virides, I will come to the definition part of that. Virides means they are very small, single-stranded, circular, low molecular weight, naked RNAs. As I mentioned, viruses means they have a nucleic acid externally surrounded by a protein coat. So we refer to as virus. But in case of viruses, the externally surrounded by protein coat is completely absent. So that's why we use the naked RNAs, and sequences ranges from just 250 to 450 bases, maximum 500 bases, which are able to cause diseases in plants are referred to as virids. So therefore, virids are nothing but very small. How small? Just 250 to 450 bases, single-stranded. Of course, viruses, RNA, all virus single-stranded, and because of complementarity in nature, they become circularized. So they take the shape of a circular ring-like structure, and they are able to cause diseases in plant. So these are referred to as the virids. The name virid derived from Latin word virid means vir means virus, id means like. Okay, virus-like particle, but it is not virus. Okay, because the externally surrounded by protein coat is completely absent in case of virid. Naked, in otherwise they are called naked plant-specific infectious RNA, tenfold much smaller than smallest known viral RNA, is called virid. These virids are regarded as the lowest step of the biological scale. So far, we believed that viruses were considered as the lowest step in the biological scale, but now these virids are regarded as the lowest step in the biological scale, molecular parasites at the frontiers of life. What are the important characteristic features of virids? As I mentioned, the virids are very small circular RNA without protein coat. This is very, very important. In case of virus, we will have protein coat and lipoprotein membrane. So both protein coat and lipoprotein membrane completely absent. Here, it is just a small circular RNA. They lack ability to code for proteins. They do not code for any proteins, unlike viruses. Viruses do code for certain proteins, certain enzymes, but here they lack genes for coding proteins. Then apart from that, these virids sometimes act like a ribozyme. That means catalytic activity. They are able to replicate themselves. So that is the very interesting part of the virid. Catalytic activity, otherwise called ribozyme activity. So these are the diagnostic features by which we can differentiate virus from virids. There was a serious disease. Why I given definition earlier, a little bit information about potato as one of the important vegetable crop. Today, the potato is one of the world staple food crop all over the world. Potato, in 1922, there was a disease called potato spindle tuber disease. It was first appeared in 1922 in commercial varieties of America, in US, and seriously, this has resulted, seriously affected the breeding program materials in US, and until 1967, majority of the plant pathologists believed that the potato spindle tuber disease was caused by viruses. So therefore, they were all looking for virus-like particle. Virus-like particles means nucleic acid, RNA, surrounded by the protein coat, but they were all unsuccessful. Of course, it was in the year 1971, T.O. Daner, a well-known plant pathologist at the US Department of Agriculture, who was working on potato spindle tuber disease, finally isolated a small piece of RNA, infectious RNA, which was responsible for causing disease in potato spindle tuber disease in potato. Therefore, he identified the causative agent of potato as responsible for the potato spindle tuber disease, and to date there are 40 different types of virids and more than 160 different types of variants have been detected in many of the agriculturally and horticulturally important crop plants all over the world. So therefore, the credit goes to T.O. Daner, who also coined the term virid, virus-like particle associated with potato spindle tuber disease, which was affecting the breeding program in America. With a lot of difficulties, he was unable to convince the world scientists that only small piece of RNA able to cause the disease in plants. But finally, with his very clear and scientific experiment, he proved that the infectious RNA, small piece of infectious RNA, able to cause diseases in potato, and he presented his paper in International Congress of Virology, and the Congress president, Andrew Lowe, one of the most respected virologists at that time, he only uttered the word, the time has come for virus. Since 1971, we have many research work has been carried out on different crop plants to identify the association of small RNAs which are able to cause plant diseases in different types of crop plants. I will go a little further into the details of potato spindle tuber virus, which was identified by T.O. Dainer in 1977. Potato spindle tuber virus has been first independently replicating subviral pathogen identified by T.O. Dainer, and this is just a electrographic picture of PSTV, potato spindle tuber virus. This is just to show the size of how small potato spindle tuber virus when compared to T7 DNA, a bacteriophage DNA. When it comes to genomes of virids, as I mentioned, virids are very extremely small. Virid genomes are extremely small in nature and covalently circular. Because of complementarity in nature, they are very small. Genomes adopt a rod-like secondary structure in vitro. The moment you isolate them, they take the shape of a rod-like structure. Okay, and this rod-like structure of virid contains 5 functional domains have been identified. So this is the typical structure of a virid. It is a single-stranded RNA, small RNA, about 250 to 450 base pairs. So because of similarity in sequences, it will take the shape of a circular in nature and become circularized, and the entire structure looks like a small rod. As I mentioned, if you go further, the details of the virid, each virid consists of 5 structural and functional domains have been identified. This includes the central conserved region. It is called CCCR region, central conserved region. Okay, and on either side of the central conserved region, we have a pathogenicity region and we have a variability region. Pathogenicity region considered to be responsible for causing disease in plant, and variability gene is the only region where each and every virids are different from each other. On either side of the pathogenicity and variability region, we have left terminal end and the right terminal end. So these are the 5 domains have been identified. In all types of virid that have been described. These virids can be classified into 2 basic categories. One which was isolated from potato spindle tuber virid disease, therefore they are referred to as family Paspiviridae, and in which the virids will be rod-shaped in structure with central conserved region. In the other type, the virid takes the shape of a hammerhead. So that's why it is called Avicen viridae or hammerhead-shaped virids. So these are the 2 types of virid that have been recognized among the 40 different types of virid that have been described so far. So these are the different types of virids have been isolated. 40 different types of virids have been isolated. Infectious RNAs have been isolated, which are known to cause diseases in agriculturally and horticulturally important crop plant. These are the important diseases caused by virids: potato spindle tuber disease, PSTVD, coconut kadang virid, citrus exocartis disease, chrysanthemum chlorotic mottle disease, chrysanthemum stunt virid, avocado sun blotch disease, peach latent mosaic disease, half stunt disease, avocado sun blotch disease, grapevine yellow speck virid. So I am just mentioned only few virid diseases. Then how to detect these infectious RNAs? As I mentioned, these are all very small RNAs, very difficult to detect. So one is by morphological methods that is based on symptomatology, but it is very difficult. Only expert can identify whether the disease is caused by viroids or whether it is disease is caused by virus or phytoplasma, because the symptoms almost superimpose with each other. So therefore, morphological method very difficult to identify viral diseases from virus and mycoplasmal diseases. DoS used a simple spectrophotometric analysis, RNA analysis, to identify the viral RNA from viroid RNA. So that method is still a very old method, but still we can rely upon that. But later, electron microscopic observations, we can also use electron microscopic observation to identify the viroid diseases. And of course, bioassay method, isolating the infectious RNAs from the infected plant and inoculating them onto a indicator plant or a healthy plant, indicator plant, we can identify whether the viroids are associated with particular crop plants or not. Later, slot blot hybridization technique using probes, RNA probes have been used, but later two-dimensional polyacrylamide gel electrophoresis was also used to separate the small RNAs from other host RNA, mRNA, tRNA, and other ribosomal RNA. But today, now RT-PCR, reverse transcription polymerase chain reaction method, most conveniently used method for detection of viroids. We have specific primers are available. RT-PCR methods can be used to detect viroids in any crop plants. And of course, once you detect them, cDNA cloning and sequencing results will help us to completely understand the structure of secondary virus, secondary nature of the RNA molecule. Conducting bioassay on indicator plant is one of the important methods. Of course, the same method we use for viruses also, but the symptoms will appear after 30 days. Look at the picture. So right one shows the healthy plant. The other 3-4 parts are inoculated with viroid taken from the infected plant. They will show you very short, short internodal regions. As a result, the growth of the plant gets affected. Leaves become very small, and growth and reproduction is completely affected. And thereby, we can within 30 days the symptoms will appear. So thereby we can test whether viroids are associated with the sample or not. Cucumis sativus, a cucumber, Lycopersicum esculentum, that is tomato plant, and tobacco, Nicotiana benthamiana. These are the most important indicator plant, bioassay plant used for the detection of viroids. How do these viroids replicate? Unlike viruses, viroids, as I mentioned, they do not code for any proteins. So therefore, they use the RNA polymerase, host RNA polymerase 2 enzyme, the host cell enzyme normally associated with synthesis of mRNA. The same RNA polymerase 2 will be utilized. The RNA polymerase 2 synthesizes the new RNA using the viroid RNA as the template. And some viroids, as I mentioned, we have rod-shaped viroid, we have hammer-head-shaped viroid, hammerhead-shaped viroid. They do possess the catalytic property, that is self-cleaving catalytic property. Therefore, the self-cleavage occurs and multiplication takes place. How do they replicate? As I mentioned, the plus strand of the viroid, once it enters into the plant system, it serves as a template, and the nucleus encoded polymerase 2 enzyme synthesizes the new strand of RNA, and this new strand of RNA undergoes linear strand of RNA undergoes circularization with the help of DNA ligase, and it becomes circularized. And the product is called the minus strand of RNA, the viroid, and this will serve as a template for the synthesis of plus strand of RNA. Again, nucleus encoded polymerase enzyme 2 play a very important role, and then again a linear plus strand linear RNA is produced, and this undergoes circularization again, and it becomes plus stranded viroid molecule. And this plus stranded viroid molecule it is the same, and circulates in the phloem cells of the crop plants, and it becomes systemic and spread to the entire crop plants. Very little is known about mechanism by which virus cause diseases in plant. Though these are the small RNAs, how do they interfere with the host metabolism? How do they take control of the entire physiological and genetic system of the host system is not yet understood. But cell wall, but once the infectious RNA, once they are present inside the plant system, they do cause cell wall malformation, membranous body formation, distorted organization, chlorotic disturbances at the cellular level, changes in the expression of pathogenesis related protein, changes in the expression of plant hormones, resulting in abnormal production of flowering and fruit setting, and also at the biochemical level. But the exact mechanism not yet understood. How do these induce disease in crop plants? Some viruses cause no detectable symptoms in their host plant, though they are present inside the plant system. Significant damages they do not cause. But there are some viruses, the moment they get enter into the plant system, they do cause damage, and it is a mysterious. It appears that the viral RNA, once it enters into the plant system, it is or its replicative intermediates trigger the RNA interference, something like a gene silencing, okay? Gene silencing, where the functional genes of the crop plant gets affected, therefore their physiology, metabolism, hormonal metabolism, everything gets disturbed, and therefore they decrease the expression of multiple plant genes, hence causing different types of disease symptoms in crop plants. So that's why disruption in cellular function is attributed to the gene silencing. Once the viral RNA enters, integrates with the plant genome, it produces the double-stranded, mRNA, the functional gene, the RNA that is produced, viral RNA, binds with the mRNA of the host, and the double-stranded RNA gets sliced down, resulting in the degraded mRNA, and therefore the resulting no functional proteins are produced or enzymes are produced that results in appearance of the disease. So gene silencing mechanism has been proposed to this how viruses cause diseases in plants. Once the viruses are produced, they move from cell to cell through plasmodesmata connections, and they traffic long distance through phloem. All these processes are associated with host proteins, and it has been observed that the plus strand of the viroids are known to colonize only in the nucleolus, but minus strands of the viroid are known to be present in the nucleoplasm. There are many different types of host range of viroids, includes both monocots. So studies all over the world proved that the viroids are known to cause diseases in both monocotyledon plant and dicotyledonous crop plant. Also, many ornamentals, vegetables are also affected by these both rod-shaped viroid as well as hammerhead-shaped viroids. These viroids have been distributed all over the world, including India, and I will be talking about little bit more about some of the important diseases. How do these viroids spread then? So it has been proposed that vegetative propagation units such as tubers, infected cuttings, and other saplings that we use for propagation purpose are likely to transmit the disease viroids. Apart from that, mechanical transmission has been proposed, and agricultural implements that we use in the agriculture fields also, if they gets contaminated, there is a possibility that they are likely to spread the viroid from infected plant to healthy plants. Infected seeds and pollens do contain the viroids, and therefore these viroids are likely to be transmitted from infected seeds and also pollens. Since viroids are phloem inhabiting molecules, phloem sucking aphids, which are likely to transmit the viroids, have been also proposed from vegetative propagation, mechanical transmission, transmission by pollen grains and seeds are the important methods by which virus spread from infected plant to healthy plant. Then how to manage these viral diseases? So planting only viral-free nuts, viral-free seeds is the only method at the present, and use of resistant varieties. So proving viral-free plant is again we need a powerful tools to identify the virus in infected plant, infected seed material. So therefore, a powerful molecular biology tools will help us. Eradication of the infected plants and trees is another important method. So phytosanitation. If you find any infected plant, just uproot them, burn them, and destroy the inoculum there itself, so that we can prevent the entry of new inoculum into the new agriculture field. As I mentioned, the phloem sucking insects are known to spread the disease. So insect control methods have been suggested to reduce the incidence. And of course, very important one is the phytosanitation method. Now here afterwards, I will take some of the important viral diseases, how they are very important and they are affecting our well-being. Look at this particular picture. It is called coconut kadang-kadang disease. And this particular picture was taken from Philippines. Philippines was once known for wonderful coconut gardens, and coconut kadang-kadang disease is a disease of coconut, Cocos nucifera. And this particular disease was reported in the Philippines in 1927 to 28. By 1962, all the 25 million palms, coconut palms have been died. So far, 40 million coconut palms have been killed since it has been discovered. So economy, once upon a time, Philippines dependent on coconut gardens for their economy. Now the economy of the Philippines was completely jeopardized. Now the country like Philippines now embarking on ecotourism, on which it is surviving. Look at the damage it has caused. Okay, the moment the disease appears, all the leaves of the coconut plant will become yellowish, and finally the drying starts, and finally dying. Kadang means drying and dying disease. It is nothing was known about the causative agent. The causative agent was detected only in 1981. Genome was isolated, and the first report appeared in Nature journal, and the causative agent, infectious RNA, was isolated, and it was about 246 nucleotide molecule, and it has central conserved region, CCR region of 44 nucleotide, and it has been renamed as CCCVD, coconut kadang-kadang viride. This is how the virides are described. Coconut kadang-kadang viride. It has number of molecular forms, and this is a purified coconut kadang-kadang RNA preparation. The RNA was isolated from the infected plant. And look at the small circular RNAs, electron micrographic picture of showing just a small RNA preparation, circularized form of RNA you can make out. So this is a CCCVD. And similarly, in the Tinangaja region of another coconut disease was discovered later, and this was known as CTIVD, coconut Tinangaja viride. It has been renamed as CTIVD, and this is one of the another important coconut. It has damaged the coconut cultivation. Potato spindle tuber viride. So as I mentioned, this was one of the earliest viride disease, and first viride was isolated from potato spindle tuber disease, where potato tubers are going to become small spindle shaped. So that is one of the important characteristic feature, and it was later named as PSTVD, potato spindle tuber viride. Look at just compare the potato spindle tuber viride, how it causes potato tubers to become small and spindle shaped. Hence the name potato spindle tuber disease was given. It was the credit goes to T.O. Dainer, for the first time identified the infectious RNAs responsible for the disease. The potato spindle tuber viride also entered tomato. So now PSTVD, now it has been reported on tomato also. Look at the leaf. Okay, once the crop gets infected, the plants become very small, growth will be affected. Instead of yellowing, because whenever there is a viral infection, most of the plants will show you yellowing symptoms. But whenever there is a viride infection, the plants will be greenish, where the growth gets affected and the leaves will be very small in size. And as I mentioned, flowering, fruit setting, everything gets affected and affect significantly the production of tomato cultivation all over the world. It has been, of course, PSTVD was under quarantine regulation in India. And we also import a lot of potato varieties. So therefore, PSTVD was a serious problem all over the world. Therefore, Government of India put PSTVD on quarantine regulation. Somehow, PSTVD has been, it has already entered India. Now, commercial varieties of potato and also tomato have been infected by PSTVD. Another viride, tomato chlorotic dwarf viride, has been identified. So this is another symptoms of viride which has been identified. Then we have bell pepper. This is one of the very important vegetable. Very recently, pepper chart fruit viride has been isolated on bell pepper, and it is where infected because the quality of the fruits are very, very important. Whenever there is an infection, the fruit size gets smaller and deformed fruits. This is one of the important characteristic feature. Apart from that, Indian apple scar skin viride. So in apple, the quality of the apple skin quality is very, very important. So here the apple skin gets a characteristic scars. Okay. So therefore, as a result, the marketability of the apple gets affected. So therefore, it is seriously affecting the marketability of apple. We have apple dimple fruit viride, where small dimples, okay, apple fruit with a deformed, crinkled skin, okay, no one will buy such fruits, if they are crinkled, okay. So this is again caused by the apple dimple fruit viride. Grapes is also not free from viride. So commercially important grape wines have already been infected with viride nowadays, okay. So therefore, it is a serious problem. Then in citrus fruits, it has been isolated. Citrus exocartis viride. Whenever there is a infection, the bark will peel. Okay, so the leaf curling is one of the symptoms, and bark cracking or bark splitting is another symptom of the disease, citrus exocartis viride. It has also been reported. Indian Agriculture Research also reported in Indian varieties of citrus fruits. Citrus bent leaf viride, so another serious problem in citrus fruit. Chrysanthemum, you know, that very important floricultural crops. Okay, India earns a lot of foreign exchequer, but today some of the varieties of chrysanthemum are affected by chrysanthemum chlorotic dwarf viride and also chrysanthemum stunt viride, where the infected plants will be very stunted in growth and they never reproduce. Beautiful flowers will not be produced at all. So therefore, the floriculture is getting affected by infection by chrysanthemum stunt viride. Columina latent viride is another important viride that has been reported. Then avocado sun blotch viride, wherein hammerhead shaped viride has been reported, avocado. And we have peach latent mosaic viride on peach. And we have cucumber is also getting infected, cucumber mosaic viride. And we have, you must have seen this plant, coleus plant, a beautiful ornamental plant. It has been reported with coleus bloomy viride very recently. So many people grow this particular plant for its beautiful variegation. But now this particular plant is known to be infected by coleus bloomy viride. You know that hop is very important, humulus lupulus. Primarily, it is used as a flavoring agent and also stabilizing agent in beer. Particularly, female strobilus of this flowers of this particular plant contains very important chemicals, and they are very important in giving characteristic flavor and taste to the beer. But very recently, the hop plants are getting infected by hop stunt virus, infecting the hop plant in Slovenia and other parts of Europe. And this is a serious problem in Europe and other parts of the world. Very recently, a HSVD hop stunt virus has been reported in mulberry. So moriculture is an important, Morasalva, very important in moriculture, where the leaf is used to feed the silkworms. So here the leaf gets affected, so affecting the moriculture, and therefore these are all some of the important representative examples how the virus, infectious RNAs affecting agriculturally important, horticulturally important, and also important cash crops in the world and also in India. So by this time, you must be asking, is there any virus infecting the animals? Yes, there is a virus called hepatitis delta. Hepatitis delta virus, which infects humans, and it has many virus-like properties. HDV has been circular RNA, genome, small genome size. HDV is also often called virus of the animals. But interestingly, HDV encodes one viral protein. Most of the virus do not code for any protein. So, but HDV is different from virus in having it code for the viral protein. Apart from that, all viruses replicate independently, by using host polymerase enzyme. But here, the HDV virus, helper virus, it needs a helper virus for self-assembly. So in that category, the hepatitis delta virus, which is earlier considered as the virus of animals, but it is not so. Another one or two points about how these, RNAs, infectious RNAs have originated. So there are different theories have been proposed to explain the origin of virus, infectious RNAs. So some believe that they are primitive viruses still surviving without any biosynthetic machinery. So they dependent on host machinery for the replication. So they are considered as relics of the RNA world, But some another school of people, they think that they are the degenerative viruses. So they developed from the low molecular weight RNAs in the past, and they are still surviving, infecting crop plants. But some others believed that nuclear RNAs might have escaped from the eukaryotic cells. Nuclear RNAs escaped from the eukaryotic cell and become pathogenic by mutation, and they are causing infection in the plant system. But however, the T.O. Dainer in 1889, he was the one who identified the viruses, hypothesized that viruses may represent living relics from a hypothetical ancient and non-cellular RNA world before the evolution of DNA or the protein. Many believe that in the hypothesis, he also thinks that the present day viruses which are infecting crop plant, they are nothing but the relics of the RNA world that exist much before the evolution of DNA or protein. So this puts our lot of theory. We have to rethink and revisit about our central dogma concept about origin of genetic material, whether DNA is the master molecule, RNA is the master molecule, or the protein is the product of RNA world. So that we need to revisit again, and therefore the discovery of the RNAs and the RNA world opened up a new era in the 21st century. Many world research scientists are working on these small, very interesting, very curious RNA molecules, and the time only will tell how important they are. Very recently, in 2022, a paper has appeared. So far, I stated that the small RNAs have been reported only in the plant system, and they are known to cause diseases in a plant. But the recent paper really, revealed that there are 400 types of small RNAs have also been isolated from animals, plants, and also crop plants. So this clearly opened up a new, dimension as far as viride research is considered. Whether I'll only specific to plant, whether they are also universally present in all other organisms, what is their exact mechanism, how do they, affect our biological process, that needs to be unearthed. So that's why virides are very, very interesting group of organisms, very curious, subviral infectious agents than viruses known to cause diseases only in the plants. So far, we believed that the virides are known to cause diseases only in plants, but the recent discoveries, the presence of small RNAs in other plant system, animal system, and also microbial system proved that they have a different role. So they come under emerging plant because these do cause diseases which come under emerging diseases. Where do they survive? Whether wild plants serve as reservoir for this virus, that needs to be discussed. Apart from that, they contain minimal genetic information, very minimal genetic information, and that's why they made them very curious agents for study. And identification of the diseases is a challenging one. Bigger, powerful tools are required for diagnosis and detection, and more studies are required to address these issues related to mode of spread, agents responsible for transmission of viral diseases, best way to manage viral diseases. That is very, very important. So they, as I mentioned, virides, the moment they enter into the plant body, so they are going to silence the functional genes. Then if they silence the functional genes, it is a serious problem. So how to identify them? How to control them? So therefore, besides classical management strategies, biotechnological approaches, including transgenic and gene silencing RNAi technology, need to be explored for controlling these viride diseases. Just to one or two slides, this is the overall presentation. So we have done some work. So therefore, I will just couple of slides for you. So how to isolate the RNAs, particularly small RNAs. So we need to isolate the total RNA from the infected plant, infected organism, where lithium chloride method, CTAB method, or trizol method can be used. Once you have done that, do the cDNA, synthesize the cDNA by using RT-PCR, convert the isolated RNA into cDNA. Once you have a cDNA, do the PCR by PCR or nested PCR using a specific primers. So for every viride, specific universal as well as specific primers are available. By using that, you can be able to detect whether the sample contains viride or not. So you will be able to get, by using PCR, you will normally get 130 base pair amplification, and then maximum 400 base amplification in the nested PCR. That means you are in the line, you are going to detect the virides. Once you do this RT-PCR, PCR and nested PCR, go with the same RNA sample, conduct a bioassay on biological sample. As I mentioned, tomato, Nicotiana, benthamiana, or cucumber. Cucumber can be used because these are the three plants which are highly susceptible. So when you inoculate by using carborundum, within 30 days, you will get a symptom. In case of viruses, within 14 or 15 days, you will get the symptoms, crinkling, yellowing. But in case of viride, you will get within 30 days, you will get a symptom. So therefore, conducting bioassay, plant bioassay and indicator plant is another method. So by using polyacrylamide gel electrophoresis, two-dimensional electrophoresis. So when you, by the same RNA sample, when you are isolating the total RNA, it contains mRNA, tRNA, ribosomal RNA, and also small RNA, viride RNA. So by doing PG, page analysis, polyacrylamide gel 2D, two-dimensional analysis, you can, isolate a small RNA very effectively. So thereby, you can isolate the RNA. So this is the infected tomato, as you know, that very important vegetable crop. It has been identified very recently that this particular plant is affected by potato spindle tuber virus. We have reported this for the first time. We used the specific phosphoviride and specific primers, and we have done this. And as I mentioned, 360 base pair amplifications means that indicates the presence of virus. We did bioassay and tomato indicator plant. We could able to amplify in all the indicator plants. We have detected the virus, and we have sequenced the genome, and we could able to construct the phylogenetic tree, and we have identified the occurrence of the natural infection of tomato by potato spindle tuber virus. It is for the first time we have detected in India, and this is the first report from our lab. Similarly, the secondary structure of the virus, potato spindle tuber virus, infecting tomato has also been deciphered from our laboratory. As I mentioned, the similarly grapevine also infected by grapevine virus has been detected, and we have doing some secondary analysis of this particular virus. Very recently, we have done work on related to mulberry. Look at this elite varieties of mulberry in Karnataka infected by obstruent virus HSVD. Okay, and we have reported again using specific primers. We have detected presence of virus in certain elite varieties of mulberry, and we have reported this for the first time obstruent virus infecting mulberry in India. So these are the work that we have carried out in our laboratory. And as you know, Louis Pasteur once said that the importance of the infinitely small is infinitely large. It is very much true with though viruses are very small infectious RNAs, they are going to cause irreversible, very significant damage both in our agricultural and horticultural crops. Therefore, it is very important. Louis Pasteur, who is considered as the discoverer of the microbial world, so long back, maybe 1860, he said that importance of the infinitely small is infinitely large. So therefore, with this quote, I will end my presentation. And if you have any specific questions, I will be very happy to answer your queries.
Hello, sir.
Yes.
Good afternoon, sir. This is Dr. Navya from Davangere University.
Yes, please tell me.
Sir, if the plant is already infected with some fungal and bacterial pathogens, will be the fate of the viroids infections?
It is called complex infection. Okay. Every infection can be detected using molecular biology tools nowadays, whether it is a fungus infected, fungal infected, bacterial infection, because specific probes are available nowadays. Okay. Yes. So from a sample which contains DNA, RNA, different, the total RNA or total DNA, from a pool of nucleic acid, you can detect whether virus is there or not by using a universal specific probes are available for exclusively for virus. Okay, so you do the general PCR. General PCR will give you amplification size of 200 base pairs. Okay, so that is an indication that virus is likely to be present. But for example, fungus. So the ITS region means you have to go around 400 or 500 base pairs. Okay, so therefore it is a small RNA. So less than 200 base pairs get amplified in the first PCR. So that clearly indicates that virus is associated.
Okay, sir. Thank you, sir.
Thank you.
Hello, sir. Thank you, sir. Hello,
yes,
sir.
Please tell me.
Myself is Dr. Venkatesh, Davangere University, sir.
Really, Venkatesh.
Sir, my question is, doubt is regarding about when we are go for a disease-free plant. So we will prefer for meristematic region. Is there any interaction between the meristematic tissue and then viral infection? Usually when the virus is infected to the plant, even though we took a expand for culturing in tissue culture, that is for only meristematic region. So is there any interaction between the virus and then meristematic tissue? We see having a more
meristematic tissue is very actively growing region.
Yes, sir.
And normally it will not have any, certain phloem because virus, virids, phytoplasma, they are all phloem inhabitating.
Yes, sir. Yes, sir.
Phloem cells are completely absent in the meristematic tissue.
Okay.
That is why they are free from virids,
Virids,
phytoplasma. Okay. Because
Okay.
all these virids, phytoplasma virids survive only in the phloem tissue, sieve tube.
Phloem tissue. Okay,
And
sir.
spread from cell to cell through plasmodesmata connection. So
Okay,
therefore, they
sir.
are absent in meristematic actively growing cells.
Sir.
So therefore, you cannot expect virids or virus to be there. That's why the meristematic tissue, meristem culture is very important tool to produce virus-free or disease-free plants.
Okay, sir.
Yes.
Thank you, sir. Thank you very much.
Thank you. Any other questions? Hope you have understood something which is something unique, this presentation, infectious RNAs. Any questions?